In this lesson, we are going to look at the linear pathway of photosynthesis. In our next lesson, we will briefly look at a different way that photosynthesis can occur in a cyclic pathway. So let's introduce all of the players in the pathway. So we will look at each individual player, and then I have an animation that's going to show how they work together. So first we have photosystem 2, and remember from our last lesson, photosystem 2 comes first in the photosynthesis pathway, but it was discovered second, so that's why it is photosystem 2. This contains the chlorophyll A, P680, which best absorbs light at the 680 nanometer wavelength. Next, we have plastoquinone. Almost had the other one right here, so PQ. It is going to shuttle electrons from the photosystem 2 and bring it to the cytochrome complex, sometimes called Site C, CYT-C. This is going to receive electrons from plastoquinone and bring them to plastocyanin, sometimes just PC. And again, this is going to shuttle electrons to photosystem 1. And photosystem 1 looks a lot like photosystem 2, and we reviewed in the last lesson what they both look like and a little bit about what they do. And they essentially do the same thing for the most part. And we're going to look in the animation on what that is. The difference between photosystem 1 and 2 is photosystem 1 has a chlorophyll A that best absorbs light at P700, at 700 nanometers, so it's called P700. Electrons are then sent to ferredoxin, which is an iron sulfur containing protein, and it's going to take electrons from photosystem 1 and send them to NADP plus reductase. NADP is a lot like NAD, so just keeping in mind that it is an electron carrier in the same sense that NAD and NADH are in cellular respiration. And so this NADP plus reductase is going to take 2 electrons to reduce NADP to NADPH. And we know that ATP synthase is going to help produce ATP through chemiosmosis. So we are going to go through this animation together and look at the entire linear pathway in an animation, and then we have the note that follows. So we have light that is going to strike one of these accessory pigments, if you remember from last lesson, and energy gets transferred, not electrons, from all of these accessory pigments to P680, which is the chlorophyll A, which we know is going to get its electron energized and actually donate its electron to the primary electron acceptor. So this P680 star represents P680 that has been energized by the photon of light. So that's what that little asterisk is. And so it gets an electron that is energized, and it is donated to the primary electron acceptor. So P680 now is going to have a positive charge because it was oxidized, it lost an electron, and the primary acceptor is going to have a negative charge. At the same time, P680 plus is going to be so electronegative that it can actually split a water molecule. So it's going to rip its electrons right from the water molecule, shown here, and it's going to give you 2 hydrogen ions and, again, half an O2 molecule, i.e. just an oxygen. And so the P680 plus takes an electron because, again, it is so electronegative. And it takes the electron, and now it is back to just being P680 because it is no longer has that positive charge because it took the electron. Essentially, this is going to keep happening. So the primary electron acceptor donates its electron to plastoquinone, and it is no longer negatively charged at the same time. P680 is going to get energized again. And its electron is going to get excited. So now it's P680 asterisk, P680 star, and it donates its electron again to the primary electron acceptor. So this keeps happening, but we're just going to look at it happening twice to work through one water molecule. And so now that primary electron acceptor is negative and P680 is positive again because it's lost its electron. So what does it do? It takes that second electron from the water molecule. And we see this movement happening. So now it's back to just being P680. So that's back to its baseline. And then we're just going to go ahead and watch the movement of the remainder of the electrons. So we have, again, the movement from P680 to the primary electron acceptor. And P680 becomes positively charged again. And it is ready to take on more electrons. But we're going to pause there. Otherwise, we could be doing this all day. So we will follow these electrons here. So plastoquinone not only shuttles electrons from the photosystem 2 to the cytochrome complex, but it is also helping build up a proton gradient. So it's going to be taking hydrogen ions from the stroma and dumping them into the lumen space. So this slide is advanced a little bit. So now we have this buildup of hydrogen ions, just so you get an idea how we have that concentration difference. And then we have plastoquinone is going to drop off its electron to the cytochrome complex and go back and get the other electron or another electron from the primary electron acceptor. And that, again, will keep happening, but we are just going to watch these electrons as we go. So the cytochrome complex is going to give its electron to plastocyanin, and again, PQ gives its electron to cytochrome complex, and we're going to see the movement of electrons through this cycle. We do see a pause here. So P700 isn't electronegative, it doesn't have a positive charge, so it isn't actually going to just take an electron in this flow. What needs to happen is another photon of light needs to be absorbed. And so we have this energy movement through the accessory pigments. We get our energized P700 to a P700 star, where it's going to actually donate its electron to the primary electron acceptor. And then it is ready to pick up another electron because it is P700 plus now. So it has a positive charge. Now it's ready to take that electron from PC, which it does right here. And of course, at the same time, we see the primary electron acceptor has a negative charge because it took that electron, so it was reduced. So now P700 is now just back to P700. It's no longer positively charged because it has that electron. And we're going to see the electron move from the primary electron acceptor to ferredoxin. And so it is back to its neutral charge. We're going to watch this happen with the other electron as well. Another photon of light has to happen. So we're at 4 photons of light. 2 electrons have to get 2 photons of light each. Once at PS2, and then once at PS1, twice at PS1. So P700 is energized, and its electron gets donated to the primary electron acceptor, reducing its charge and making the P700 have a positive charge. Now it's ready to take that second electron. Okay, and we see again the ferredoxin is giving its electron, and we just see the electrons move through the path. So PC gives its electron to P700. Now P700 again is back to baseline. Now this electron is going to leave the primary electron acceptor, and again this would be continuous. We would see more than just two electrons moving through the system at once. And we aren't going to see the electron go to ATP synthase. We know that's not what happens. What we do see is the conversion of NADP to NADPH. So watching this, now we have two electrons here at the NADP plus reductase, and we're going to have NADP plus come in with two other hydrogen ions, and we're going to get NADPH. So we've had a removal of a hydrogen ion. So again, we're getting less hydrogen ions in the stroma, and at the same time, we're getting hydrogen ions dumped into the lumen from the stroma. So again, creating a concentration gradient between the two sides of the membrane. Okay, and then we know what's going to happen with the ATP synthase. We're going to have a whole bunch of hydrogen ions moving through the ATP synthase to create ATP. And we know ADP plus inorganic phosphate is going to be giving us our energy source of ATP. So let's look at some note filling in for you guys. So photosystem 2 came first, and this is where we have the splitting of water into electrons. And we know it's also creating protons, which adds to that proton concentration in the lumen side. And we also get oxygen gas created. When the photon of light strikes the antenna complex, so that whole protein structure embedded in the membrane with all of the accessory pigments, the absorbed energy is transferred to P680, and the electron gets energized, and we get P680 star. The excited electron is then transferred to the primary electron acceptor molecule, and that becomes negatively charged, so it was reduced. P680 now carries a positive charge, so it was oxidized. And then now, as P680 plus, it is now so electronegative that it can actually remove electrons from the water. So that's extremely fascinating, if you will. So reduction of P680 plus to P680 is facilitated by the water splitting complex, which is on the lumen side of PS2. So it's driven by strong electronegativity, which allows the water to be oxidized, the two electrons being taken. So one electron we saw happening at a time is given to P680 plus to make it neutral again. The acceptor molecule, A, in our diagram, got its electron from P680, and it transfers its electron to plastoquinone, PQ. The acceptor molecule becomes neutral again, allowing this whole process to start over. And note, for every water molecule, this process happens twice, which we saw in our animation. So our animation was actually a little simplified because it's actually the water splitting complex will bind two water molecules at once, but we looked at just one water molecule, and we saw that from one molecule of water, you were getting two electrons taken. So again, we're looking only at linear electron transport today, and then our next lesson we'll look at a little different scenario where we have cyclic electron transport. So we have the oxidation of P680 where it lost electrons and became P680 plus. It transferred its energy, high energy electron to the primary electron acceptor, and it's actually called pheophytin. But the textbook doesn't talk about that, and I'm not going to make you remember that because you have enough to remember. The oxidation reduction of plastoquinone happens next, where the electrons are transferred from the acceptor molecule to PQ, and PQ will move through the lipid bilayer and at the same time picks up hydrogen ions and dumps them into the lumen. And then it donates its electrons to cytochrome complex and, as I mentioned, releases that hydrogen ion into the lumen. So again, this is increasing that proton concentration gradient between the stroma and the lumen. Next, the electrons are transferred from the cytochrome complex to plastocyanin. We then have another double photon usage where for each electron transferred from plastocyanin to the photosystem 1, we need another activation from the photon of light to energize P700 to become P700 star. It'll then transfer electrons to the primary electron acceptor of photosystem 1. It becomes positively charged, P700 plus. And now, because it is positively charged, it will accept an electron and be reduced back to P700 by plastocyanin. You need to be practicing following the electron and explaining the reduction and oxidation process. So next, electrons transfer to NADP plus by ferredoxin. So the electrons from P700 are transferred to the ferredoxin, and this again is an iron sulfur protein. The oxidation of ferredoxin leads to the transfer of electrons to NADP plus, and again it's going to be 2 electrons being transferred to give us NADPH. So first NADP and then NADPH, and again removal of a hydrogen ion from the stroma occurs as NADPH is being produced. This NADPH is going to behave a lot like NADH, and we'll see that a little bit further on. So lastly, we have chemiosmosis in this electron transport chain happening, where the whole point is to get this concentration gradient occurring, and we know that we can get the synthesis of ATP as hydrogen ions pass through the ATP synthase. So protons are allowed to pass through the pores in the ATP synthase, and we know the ATP synthase are embedded in the membrane, and this is the thylakoid membrane that it's embedded in. So it's the same kind of business as cellular respiration. As protons move through, ATP is synthesized from ADP and inorganic phosphate. So just looking at energy levels as a visual, in cellular respiration, electrons are pulled from high energy NADH to low energy oxygen. In photosynthesis, we are actually seeing the opposite. We start with a low energy water molecule and we end up with a high energy NADPH. So we need the light energy to drive the low energy electrons from water. So that's the whole beauty of the photon of light. They're giving energy to a source that didn't have much energy to begin with. And so we see this kind of Z pattern of energy. So low energy, we get zapped. High energy electrons, as they move, they're losing electron, and they get zapped again at photosystem 1. So they have high energy again, and then that energy is stored as NADPH. So in summary, each water molecule split produces 2 electrons. So for each water molecule split, you are getting 2 electrons moving through. Each electron needs a photon of light at both photosystems. So one electron needs 2 photons of light, one at photosystem 2, and then again at photosystem 1. So we have 2 electrons, and they both need 2 photons of light, so we're at 4 photons of light. Now, as I mentioned earlier, I planted a little seed that PS2 binds 2 water molecules at once. So we're getting 2 water molecules, so we're getting 4 electrons, which means we need 8 photons of light for one whole linear pathway, if you will. 2 water molecules are going to release 2 electrons each, so we're at 4 electrons. Each electron needs 2 photons of light. So 4 times 2 is 8 photons of light. Thank goodness there's so much light. And here is our overall reaction. We have 2 water molecules. Again, the water splitting complex binds 2 water molecules at once. We have 4 hydrogen ions being released, 4 electrons that are going to get pumped through the pathway, and a full water oxygen molecule created. So that's our linear photosynthesis. Please spend some time with it. We are going to be practicing explaining it to our peers and with me to make sure we understand the overall process, and practicing explaining the oxidation reduction that's happening, and understanding how many electrons and photons of light and all of the important things that happened.